In human genetics, a human Y-chromosome DNA haplogroup is a haplogroup defined by differences in the non-recombining portions of DNA from the Y chromosome. It represents human genetic diversity based on single-nucleotide polymorphisms on the Y chromosome. Y-DNA haplogroups represent major branches of the Y chromosome phylogenetic tree. Y-chromosomal atom is the name given by researchers to the patrilineal most recent common ancestor of all living humans at the root of this tree. Estimates of the date when Y-chromosomal atom lived have varied significantly in different studies. Archaeological and genetic data suggest that the source populations of Paleolithic humans survived the glacial maxima, and human Y-DNA haplogroups emerged in sparsely wooded refugia, and dispersed through areas of high primary productivity while avoiding dense forest cover. Naming convention. Y-DNA haplogroups are defined by the presence of a series of Y-DNA SNP markers. Subclades are defined by terminal SNP, the SNP furthest down in the Y chromosome phylogenetic tree. The Y chromosome consortium developed a system of naming major Y-DNA haplogroups with the capital letters A through T, with further subclades named using numbers and lowercase letters. YCC shorthand nomenclature names Y-DNA haplogroups and the subclades with the first letter of the major Y-DNA haplogroup followed by a dash and the name of the defining terminal SNP. Y-DNA haplogroup nomenclature is changing over time to accommodate the increasing number of SNPs being discovered and tested, and the resulting expansion of the Y chromosome phylogenetic tree. This change in nomenclature has resulted in inconsistent nomenclature being used in different sources. This inconsistency, and increasingly cumbersome longhand nomenclature, has prompted a move towards using the simpler shorthand nomenclature. In September 2012, Family Tree DNA provided the following explanation of its changing Y-DNA haplogroup nomenclature to individual customers on the Y-DNA results pages. Major Y-DNA haplogroups. Major Y chromosome haplogroups include tree view groups A and B. Haplogroup A is the African macrohaplogroup from which all modern haplogroups descend. BT is a subclade of haplogroup A, more precisely of A1b, as follows: haplogroup A0, haplogroup A1, A1A, A1B, A1B1, A1A, A1B1B, BT, haplogroup BCT. Groups with mutation M168. The defining mutations separating CT are M168 and M294. These mutations predate the out-of-Africa migration. The defining mutations have probably occurred in northeastern Africa some 65,000 years ago. The P143 mutation that defines haplogroup CF may have occurred at that time, bringing modern humans to the southern coast of Asia. Haplogroup CF found outside of Africa, throughout Eurasia, Oceania, and the Americas. Haplogroup C found in Asia, Oceania, and North America. Haplogroup C1, haplogroup C1 or haplogroup C1A1 found with low frequency in Japan. Haplogroup C1A2 found with low frequency in Europe and Nepal. Haplogroup C1b, haplogroup C1b1, haplogroup C1b1a, haplogroup C1b1a1 found with low frequency in the Indian subcontinent, the Arabian Peninsula, and northern China. Haplogroup C1b2, haplogroup C1b2a found in Indonesia, New Guinea, Melanesia, Micronesia, and Polynesia. Haplogroup C1b2b found among the indigenous peoples in Australia. Haplogroup C2 found throughout Eurasia and North America, but especially among Mongols, Kazakhs, Tungusic peoples, Paleo-Siberians, and Na-Dene-speaking peoples. Haplogroup F. Haplogroup de California. 65Ca Haplogroup D found in Japan, China, the Andaman Islands. D1 D1a Haplogroup D1a1 found mainly in Tibetans, Qiangic peoples, Yi, and Hmong-Mien peoples. Haplogroup E1b Haplogroup E1b1, formerly E3 Haplogroup E1b1a found in Africa, especially in Niger-Congo speakers, formerly E3a. Haplogroup E1b1b found in East Africa, North Africa, the Middle East, and Europe, formerly E3b. Haplogroup E2. Groups descended from Haplogroup F. The groups descending from Haplogroup F are found in some 90% of the world's population, but almost exclusively outside of sub-Saharan Africa. The mutation of IJ corresponds to a wave of migration out of the Middle East to South Asia of some 45Ca that subsequently spread into Europe. Haplogroup G originated in the Middle East or perhaps further east as far as Wardak region of Afghanistan, some 30Ca, and spread to Europe with the Neolithic Revolution. Haplogroup H probably occurred in India some 30 to 40Ca, and remains prevalent there, spreading westwards in historical times with the Romani migration. Haplogroup K spread widely to Eurasia, Australia and the South Pacific. Paragroup F** found in southern India, Sri Lanka, Yunnan, Korea. Haplogroup G California, 21Ca found in many ethnic groups in Eurasia, most common in the Caucasus, Iran, Anatolia and the Levant, found in almost all European countries, but most common in Gagauzia, southeastern Romania, Greece, Italy, Spain, Portugal, Tyrol, and Bohemia, with highest concentrations on some Mediterranean islands, uncommon in northern Europe. Found in small numbers in northwestern China and India, Pakistan, Sri Lanka, Malaysia and North Africa. Haplogroup G2A3 Haplogroup G2A3A Haplogroup G2A3B Haplogroup G2A3B1 Haplogroup G2B Haplogroup G2C Haplogroup G2C1 Haplogroup H2 Haplogroup IJK Haplogroup IJ California, 45Ca Haplogroup I found in Europe and parts of the Near East. Haplogroup I1 found mainly in Northern Europe. Haplogroup I2 found mainly in Southeast Europe and Sardinia save for I2b1 which is primarily found in Western, Central and Northern Europe. Haplogroup J Haplogroup J** Haplogroup J1 associated with Northeast Caucasian peoples in Dagestan and Semitic peoples in Mesopotamia, the Levant, the Arabian Peninsula, Ethiopia and North Africa. Haplogroup J2 found mainly in Mesopotamia, Anatolia, Levant, Greece, the Balkans, Italy, Iran, South Central Asia and the Caucasus. Haplogroup K spread all over Eurasia, Oceania and Americas. LT groups descended from Haplogroup K. Haplogroup L is mainly found in South Asia. Haplogroup M is most prevalent in Melanesia. The No haplogroup appeared California, 35 to 40Ca in Asia. Haplogroup N probably originated in Southeast Asia and spread north into Siberia and west, being the most common group found in Uralic peoples. Haplogroup O is found at its highest frequency in East Asia and Southeast Asia, with lower frequencies in the South Pacific, Central Asia, and South Asia. Haplogroup P gave rise to groups Q and R, and is rarely found in its undifferentiated stage. It probably originated in Central Asia or the Altai region. Haplogroup Q also originated in Central Asia, migrating east to North America. Haplogroup K** found in Melanesia and Australia. Haplogroup K1 found in Indian subcontinent. Haplogroup K2, Haplogroup K3 found in Melanesia and Polynesia. Haplogroup K4 found in barley. Haplogroup LT. Haplogroup L found in South Asia, Central Asia, Southwest Asia, the Mediterranean. Haplogroup T found in Africa, the Middle East, the Mediterranean, South Asia. Found in a significant minority of Syaxensi, Somalis, Avisinks, Stilfsa, Ethiopians, Fulb, Egyptians, and Omanis. Also found at low frequency throughout the Mediterranean and parts of India. Haplogroup M. NOPS. Haplogroup M found in New Guinea and Melanesia. Haplogroup number 35 to 40 KYA. Haplogroup No**. Haplogroup N found in northernmost Eurasia, especially among the Uralic peoples. Haplogroup O found in East Asia, Southeast Asia, the South Pacific. Haplogroup P. Haplogroup P**. Haplogroup Q found in Siberia and the Americas. Haplogroup R found in Europe, West Asia, Central Asia, and South Asia. Haplogroup S found in the highlands of New Guinea. Groups descended from Haplogroup No. The No haplogroup appeared California. 35 to 40 Car in Eastern Asia. Haplogroup N possibly originated in Eastern Asia and spread both west into Siberia and north, being the most common group found in some Uralic-speaking peoples. Haplogroup O is found at its highest frequency in East Asia and Southeast Asia, with lower frequencies in the South Pacific, Central Asia, and South Asia. Haplogroup number 35 to 40 Car. Haplogroup N found in northernmost Eurasia, especially among the Uralic peoples. Haplogroup N1. Haplogroup O found in East Asia, Southeast Asia, the South Pacific. Haplogroup O1. Haplogroup O1A found in eastern and southern China, Taiwan, and Southeast Asia, especially among Austronesian and Ticoidai peoples. Haplogroup O1B. Haplogroup O1B1 found in Japan, southern China, Southeast Asia, and the Indian subcontinent, especially among Austroasiatic and Ticoidai-speaking peoples, Malays, and Indonesians. Haplogroup O1B2 found in Japan, Korea, Manchuria, and Southeast Asia. Haplogroup O2 found throughout East Asia, Southeast Asia, and Australasia including Polynesia. Groups descended from Haplogroup P. Haplogroup P has two branches. They are QM242 and RM207, which share the common marker M45 in addition to at least 18 other SNPs. Haplogroup Q. Q is defined by the SNP M242. It is believed to have arisen in Central Asia approximately 17,000 to 22,000 years ago. The subclades of Haplogroup Q with the defining mutation, according to the 2008 ISOGG tree, are provided below. SS4BP RS41352448 is not represented in the ISOGG 2008 tree because it is a value foreign street. This low-frequency value has been found as a novel Q lineage in Indian populations. The 2008 ISOGG tree QQ** Q1 Q1** Q1A Q1A** Q1A1 found with low frequency among Dungans, Han Chinese, Hazaras, Japanese, Koreans, and Tibetans. Q1A2 found at low to moderate frequency among some populations of Southwest Asia, Central Asia, and Siberia. Q1A3 Q1A3* found at low frequency in Pakistan, India, and Tibet. Q1A3 atypical of indigenous peoples of the Americas. Q1A3A** Q1A3A1 found among some indigenous peoples of South America, such as the Ticuna and the Wayuu. Q1A3A2 Q1A3A3 Q1A4 Q1A5 Q1A6 found in a significant minority of Yemeni Jews. Q1B found at low frequency among samples of Hazara and Sindus. Haplogroup R. Haplogroup R is defined by the SNP M207. The bulk of Haplogroup R is represented in descendant subclade R1, which likely originated on the Eurasian steppes. R1 has two descendant subclades, R1a and R1b. R1a is associated with the Proto-Indo-Iranian and Balto-Slavic peoples, and is now found primarily in Central Asia, South Asia, and Eastern Europe. Haplogroup R1b is the dominant haplogroup of Western Europe and also found sparsely distributed among various peoples of Asia and Africa. Its subclade R1b1a2 is the haplogroup that is most commonly found among modern Western European populations, and has been associated with the Italo-Celtic and Germanic peoples. Haplogroup R1 found throughout Western Eurasia. Haplogroup R1a found in Central Asia, South Asia, and Central, Northern and Eastern Europe. Haplogroup R1b found in Western Europe, West Asia, Central Asia, North Africa, and Northern Cameroon. Haplogroup R2 found in South Asia, Caucasus, Central Asia, and Eastern Europe. Chronological development of haplogroups.